Ora si registrerà la conferenza.
So in order to understand the field of neuroscience, research in being up to date on cutting edge research and the gaps in the scientific community, we are working with key opinion leaders globally, as you can see in the short video. We are working across a wide range of disease areas, which include multiple sclerosis, for example.
It's the commonest autoimmune disease of the central nervous system.
It's an amazing time to be a glial biologist, particularly reactive astrocyte function in chronic neurodegenerative diseases like Alzheimer's disease is a fascinating topic for investigation.
There are several trends which are emerging, which are exciting as this field is moving forward.
So these are just a few examples of great collaborators that we have across the globe, but we've also been very busy throughout these last few years to develop a strong portfolio and resources across a wide range of areas with high unmet patient need. So these include neurodegenerative disorders like you've seen across Alzheimer's and Parkinson's, but also Huntington and ALS. We've also expanded to traumatic brain injury, an area with high unmet need, but also to multiple sclerosis, neuropsychiatric disorders such as schizophrenia and depression, but also more recently we've entered the area of cerebrovascular dementia, including stroke. Now, what's very exciting is that at Abcam we continue to innovate and to enter other spaces so we can support more neuroscientists. So I would highly encourage you to look at our Neuroscience Hub page, which is highlighted in the bottom of the screen, and come check for novelties in this space. Now, on the next slide, I'm just highlighting an example for Parkinson's disease of the type of resources that we've been putting together to support you with your research, and especially as you entering this space. So we've developed a very useful Parkinson's disease pathway in collaboration with key opinion leaders. We've also provided videos, overviews in this space, for example, with Professor Roger Barker at the University of Cambridge, but also Professor Angelo Antonini, University of Padova. And we've also been working with the Michael J. Fox Foundation for more than 10 years now to develop a full suite of products in this space that go all the way from rabbit monoclonal antibodies to simple stabilizers and proteins to support, again, to support your research. And really our focus, as is highlighted on the next slide, is really about being able to provide you with the full solution. And these include support you at each stage of your experiments. So for example, if you're defining your paradigm and develop your conditions for your research, you might want to look at our range of biochemicals or recombinant proteins. If you then want to look at and measure circulating levels of proteins or neuroinflammation in biological fluids such as plasma or CSF, you can look at our simple step range for singleplex or look at our Fireplex assays for multiplex applications. We also have a very large range of recombinant antibodies to provide with that consistency and reproducibility that your research requires. And we go not just looking at the total proteins, but actually allow you to look at specific isoforms, specific phosphoforms, and also have conformation-specific antibodies that, for example, can be helpful for you to look at folding of proteins such as alpha-synuclein. Now, we understand that your research requires some flexibility, and so we try to provide our products in different formats. So we also have carrier-free versions of our recombinant antibodies that you can use together with our lightning kits to do conjugation and apply for multiplexing applications. But we also have primary conjugates with a wide range of Alexa fluorophores that you can use already prepared. And finally, and very relevant for the discussion today, we also have a wide range of edited cell lines, including knockout cell lines that you can use for your functional assays, or also the human iPSC-derived cells, where you can use as human disease models for your important research and to test your hypothesis. So with that in mind, I would like to introduce you our partners at bit.bio, and my colleague Kam, who's the SVP for Alliance Management at bit.bio.
Thank you very much, Mariana, for that introduction. It's a pleasure to participate in this presentation. As you will see, all of us as scientists are aware of a common shared problem in drug discovery. And that problem very much relates to the fact that it's very challenging to access high quality human cells. Now at bit.bio, our focus is to provide consistent and scalable human iPSC cells which have been derived from iPSCs for in vitro disease modeling and for drug discovery. Now, many of you will be also aware that, sorry, could you just go back one slide? Many of you are also very much aware of the challenges involved in cell therapies. Now, with respect to the challenges both in cell therapies and drug discovery, the majority of clinical trials have primarily failed because it's very difficult to recapitulate disease biology in cell lines or in animal models when you are trying to model human disease. And so, again, even with the first CAR T cell therapy treatment, Chimera, which was back in 2017, this provides a glimpse of what is possible when we are able to address certain types of cancers. But again, the same problem arises in terms of accessing high quality human cells. Next slide, please. We're very much aware of the costs involved in drug discovery, coming up to $2 billion in order to bring a drug to the market. I think we're also very much aware of the challenges in the clinic in terms of the failure rate. Nearly 90% of clinical trials actually do not make it into phase 3, or more importantly, in CNS, the failure rate is actually much greater. If you go to the next slide, our Chief Technology Officer, John Connolly, is an expert in the cell therapy space. And in my previous discussions with John, he points out that if you want cell therapies to be accessible to the majority of patients, then we have to address the affordability question, and most importantly, resolve the issues around consistency and scalability. And it is in this environment that bit.bio operates in. And what we are aiming to do is to provide human cells to support and aid drug discovery, to impact and reduce the failure rate in the clinic, and to enable a generation of new drugs, as well as apply that same fundamental technology to address the issues and challenges associated with cell therapies, so that we can provide scalable manufacture of human cells to help generate a new generation of cell therapies. Next slide. At bit.bio, we are applying the principles of computation to biology. And by doing this, we are engineering cell types. How we do this is powered by a technology called Optiox. Optiox stands for Optimized Inducible Overexpression. It is, in effect, a genetic switch, want of a better phrase. At bit.bio, we are focused on developing a scalable technology platform that is capable of producing consistent batches of every human cell, that has applications in research, in drug discovery, in toxicology, as well as in the broader area of cell therapies. Next slide, please. if you just do one click. Now, many of you as scientists are well aware of the lost promise of iPSCs. iPSCs have been around for more than 2 decades now, and many of us have worked in the lab and developed protocols using directed differentiation, where you are manipulating the external environment of a cell, of an iPSC, to direct it towards a specific phenotype. Now, this requires manipulation in terms of the media with respect to growth factors, cytokines. Many of these protocols are not trivial. They're very challenging. There's issues around consistency, scalability, maturity, and purity. And this is the classical approach, as many of you will be aware, that you are relying on exotic media components, sometimes ill-defined, and the protocols can be seemingly very challenging to transfer into a drug discovery setting. Can you just click one forward, please? Now, the work that was done by Professor John Gurdon, as well as Yamanaka, really opened the eyes in terms of the importance of cellular reprogramming. And it was through their work, their foundational work, that we observed the role of transcription factors, which can play a key role in terms of determining the eventual phenotype of a cell. However, even in the early days of cellular reprogramming, the challenges of a heterogeneous population and low scalability were still there. If you click one more forward, please. bit.bio has taken some of these challenges and turned them on its head. We are, with the use of this Optiox technology, which in effect, once we've identified transcription factors, we can introduce these transcription factors into this genetic switch and then incorporate this into any iPSC. We are in effect hardwiring or coding, want of a better phrase, the phenotype of an iPSC to that particular cell type of interest, thereby driving purity, scalability, and consistency. Next slide, please. In this video, what you should see is this is a skeletal myocyte, a skeletal muscle that's been reprogrammed using MyoD1, a well-known transcription factor. This reprogramming is being powered by Optiox. And what I'd like to make a note of is how this is happening in a matter of days. So we're getting a very significant population of these striated, elongated skeletal myocytes that is occurring in a matter of days. And during the course of this presentation, you will see how easy these cell types are available in terms of their use. And by using Optiox, we're able to use its advantages in such a way that we can provide consistency, maturity, and speed. And if you add acetylcholine, you can see that in terms of the treatment, they lift off the plate, they contract and detach. So if you go to the next slide. Now, what I'd like to start here is really to introduce Optiox. And Optiox, I'm sat in front of a HP computer. Right in front of this is an Intel processor. Now, I want you to imagine that Optiox is like that 10th generation Intel processor. And the computer code that allows us to talk and the visualization of using GoToMeeting is really the transcription factor code. So thinking of this analogy, I want to now apply this to what we're doing here within bit.bio. We have developed a proprietary technology called Optiox. And what Optiox allows us to do is to enable precise control of any transcription factor combination. We can apply this in a scalable manner. Now, Optiox is based upon a dual targeting system based on 2 safe harbor locus. One is AAV-S1, the other is ROSA 26. We also have a co-exclusive license to use the TET third generation switch. And it's this combination of the dual targeting system and the use of these two safe harbor locus alongside the TET-on system, which allows us to incorporate the transcription factors into these safe harbor locus. You click on. Once we've identified these transcription factors, we can then, by simple inclusion into the Optiox cassette and by incorporating these randomly into an iPSC, adding doxycycline, we can then drive that cellular programming. And you will see during the course of this presentation, our first product, which is the IO glutamatergic neurons, and also the IO skeletal myocytes that we have developed. We will talk in more detail. But again, this is all about being able to produce cell types that are more biologically representative of human disease. Next slide, please. So there are two key fundamental aspects to our platform. One is the discovery platform, and the discovery platform is a high throughput combinatorial platform that is dedicated and based upon the use of machine learning algorithms to identify transcription factors. Every cell type will have a unique combination of transcription factors, and that is a process that we are applying in a very diligent way by using RNA-seq data, by using functional data in terms of genomic information that helps drive the algorithms that we have developed to generate potential transcription factors. We take those transcription factors and then incorporate them into this Optiox, this cassette. Once we have the optimal combination of transcription factors, we can then use that and program to any particular cell type that we want. And as you can see here, we have the IO glutamatergic neurons, the IO skeletal myocytes, and we are developing a whole range of other cell types, not just in CNS, but also in metabolic disease, in immune cells, and other tissue types as well. Go to the next slide. So what I wanted to finish on before I hand over to Kaiser is essentially this applies to any cell type that comes from bit.bio. First of all, the level of characterization is essential. As researchers, you want to know that do these cells accurately recapitulate a particular disease type, a particular mutation. So let me go over some of the key characteristics of our cells. First of all, rapid maturity. This is very important. We don't want basic researchers and drug discovery scientists to be waiting weeks and weeks and weeks in order to use these cells. These cells are ready to use within days upon post-revival. This differs very much from the classical approaches. We have given a lot of thought into the level of characterization, and so each of our cell types will be characterized in terms of immunocytochemistry plus gene expression data. That's invaluable information to the researchers because they want to know if a particular parental wild type has the particular gene subsets of interest to them. Furthermore, batch-to-batch reproducibility and creating a homogeneous population that can be used again in basic research and HDS. Doing this at scale. So we know our technology has scalability because we're applying the fundamentals of this technology in a completely unrelated area, in the area of artificial meat. So there we're working with a collaborator to manufacture billions and billions of cells, driving consistency, driving a homogeneous population in terms of reproducibility. Also very important to researchers is cost effectiveness and and the cells to be ready to culture. What you'll see during the course of this presentation is the number of cells that will be required in terms of per centimeter squared of a plate is much, much lower compared to our other cell types which are produced by cell differentiation methods using the classical approach. In addition, we have created two types of cells whereby, in order to minimize two types of vial sizes containing different populations of cells. So we have a small size vial and a large size vial in order to minimize waste. And finally, plate compatibility, working across 6 well plate to 384 plate formats. So at this point, I'd like to hand over to my colleague, Kaiser, who will talk in more detail about the individual cell types, and he will start with the IO glutamatergic neurons. Firstly, thank you very much.
Thank you, Cam. Thank you, everyone, for coming to this talk today. So today I would like to highlight this Optiox application at bit.bio to make 4 different cell types. The first one, as Cam has alluded to, is the IO glutamatergic neurons. These are functional human iPSC-derived glutamatergic neurons. And we launched it to the market late last year with our partner Abcam. So the cells are first primed at bit.bio so that customers receive a vial containing highly pure population of neurons. When they are thawed, they are allowed to continue their reprogramming for a further 4 days. So this video here provides a view of what that looks like. As you can see, they arrive as committed neurons and then continue to differentiate, forming these long axons that seek and form functional connections with other neurons. And we can also appreciate the high purity of the culture in this video. You can see that most of them here are neurons, actually. This can also be further illustrated in a bulk RNA-seq analysis that we did. This is a bulk RNA-seq time course of the reprogramming. This is a PCA graph of that RNA-seq data. And what it's showing you is the consistency, just how rapid and highly consistent this neuronal reprogramming is thanks to the Optiox system. And it shows this rapid, highly controlled chronological ordering of the time points. And in each time point, you have 3 replicates, and that shows you how consistent that process is. And it follows this tight time course and forming neurons as early as day 4. But by day 14 post-induction, most of them have become bona fide neurons, and you can see that they're very much differentiated from the earlier time points, indicating that they've reached a stable, mature state. We also did a single-cell RNA-seq analysis of the same time points. Here they show that they are largely glutamatergic based on this marker here, vGlut2. At day 11, you can see most of them are positive for this marker here in our single-cell RNA-seq data, and that makes up about 80% of the culture. There's a small population of cholinergic neurons, which stands for which is identified by this particular marker, but that makes up about less than 20% of the culture. More importantly, all of them are neurons, and almost more than 80% of them are glutamatergic. With our partner, we've also stained these neurons for some classic neuronal markers. You can see as early as day 1 post-induction, they're already expressing alpha-tubulin, and they've got this very specific neuronal marker called mu-n. And by day 11, they've got they stain for MAP2, MAP2 being very important for observing dendrites. And you can see many of them, you can see how they are localized in the particular dendrites. And by day 3 and day 4, by day 11 also, they express all these 2 other classic neuronal markers, tau and beta-3 tubulin. And you can see very nice high expression of these neurons, again, showing you how robustly they express these markers and how consistent, how pure the culture is. And this is by day 11 post-revival. This figure here demonstrates the maturity of our neurons through the expression of these tau isoforms. So what we know is that compared to immature neurons, mature neurons have a higher expression of this 4R isoform, and it is known to be in equal proportion to the 3R isoform. So here you can see, a day 4 after the customer receives it, they're still slightly immature. They have more 3R, 4R isoform, but you can see how that rapidly changes. And, as early as 14 days post-revival, they've already got a balanced proportion of these two isoforms, indicating that they're very much mature neurons as early as 14 days post-induction, which is, of course, very important for modeling tau-related diseases. These neurons are functional as well. They form functional neurons within 8 days post-revival and functional networks within 13 days, evidenced by synchronous bursting activity. This heat map here, we compared of multi-electrode array based electrophysiology, was recorded by our partner, Charles River, and it compares it alongside iPSC-derived cortical neurons at day 47 post-induction from iPSC, and striatal neurons at day 38, much later compared to our neurons at day 20. And you can see how, in terms of activity, how comparable and in fact how active these neurons are even at an earlier time point. This activity is largely AMPA mediated. We did some further electrophysiology study with a lab here at the LMB in Cambridge, John O'Neill's lab. And basically this figure here shows you that in the presence of CNQX, which is specific for AMPA activity, it knocks out all the synchronous bursting that we see in the control. So suggesting that our largely glutamatergic activity drives the synchronous bursting that we see in our culture. And when we wash off the CNQX, we see that this activity returns. And when we add TTX, which blocks all neuronal activity, we see that it's completely wiped out. So again, just to demonstrate that our activities, our neurons are glutamatergic and specifically AMPA-mediated glutamatergic neurons. To emphasize the utility of these cells as a cell model, our partner Charles River harnessed their remarkable properties to demonstrate its effective application for drug discovery, both using electrophysiology-based assays like the MEA data that I've showed you so far, and also using high-throughput compound screening assays. If you would like a copy of this poster with Charles River, feel free to email us, and we can provide that for you. As Kam mentioned, our products will come, for these neurons will come in 2 sizes, a smaller size and a larger size. A smaller size that can plate a minimum of half your standard 24 well plate, your standard tissue culture well plate. And the large vial can make, is enough to plate a minimum of 1 24 well plate. And with that vial, it's already been, we've gone through industry standard quality checks and has been tested for applications ranging from ICC, Western blot, 3D high throughput screening, next generation sequencing assays, and has a range of applications from academic research to neurotoxicology, genetic screening, so on and so forth. Now, the next cells that we are going to launch very soon with our partner Abcam are the human-induced skeletal myocytes. This is generated as again from human iPSCs by MyoD-driven optiox cellular reprogramming. These skeletal myocytes are available at scale and easy to culture. So this video here captures the first 10 days of reprogramming. And you can see within days of reprogramming, upon revival in the hands of the user, they form this very nice, highly pure culture of myocytes. And you can appreciate the rapid morphological changes that are occurring. By day 10 post-revival, the cells express components of the contractile apparatus with visible striated myocytes that contract in response to acetylcholine. As with the neurons, these myocytes are also highly pure, and these immunofluorescence stainings of these cells show staining at day 10 post-revival for robust expression of components of this contractile apparatus that I mentioned, like desmin, dystrophin, myosin heavy chain, along with the muscle transcription factor myogenin. The cells also demonstrate the expression of troponin with visible striated fibers and multinucleation, as you can see here, stained by DAPI, which is a very, very important feature for skeletal myocytes, this multinucleated morphology. Following reprogramming, the skeletal myocytes robustly downregulate expression of pluripotency markers, SOX2 and OCT4, and begin to express myosin heavy chain isoforms such as MYH3 and MYH8. Through continued culture, the cells demonstrate expression of mature myosin isoforms such as MYH7 and MYH1, along with desmin, dystrophin, myogenin, and titin. Critically, for metabolic studies, this data here demonstrates expression of the insulin-regulated glucose transporter, GLUT4. This RT-PCR data here shows that as early as day 10 post-revival, they demonstrate the expression of this very important marker compared to undifferentiated HiPSCs and glutamatergic neurons. And you can also see this in immunocytochemistry, again, seeing this very nice multinucleation and also staining for the GLUT4. And this is also evident in a Western blot staining as well, which makes it a very nice model for investigating metabolic diseases like insulin resistance, for instance. Another demonstrated functionality is that the cells can be used in contractility assays. Now, if you look at this video, I hope you can appreciate how these muscles are actually contracting spontaneously in the culture. So it makes it a very nice cell model for studying contractility. As with before, the myocytes will come in two sizes, a small, which also can plate half a 24-well plate, and a large vial, which can also plate an entire 24-well plate. And we have tested it for a range of applications, ranging from ICC and Western blot, and for numerous applications as well. Now, another cell on the horizon for us, which we hope to launch soon, is the human iPSC-derived GABAergic neurons. Now, these neurons express typical GABAergic markers such as GABA itself and the GABA transporter VGET1. And like the glutamatergic neurons, our technology can reprogram these cells in a matter of days with high efficiency. In this time-lapse video, you can see how these neurons emerge out of these clusters as they complete their conversion to GABAergic neurons. This form of migration as part of a differentiation process is very similar to what happens in normal development or even in classical differentiation protocols, just that we are seeing it here in this rapid, highly efficient manner. This efficiency is also observed in a single-cell RNA-seq that we did, a single-cell RNA-seq time course. You can see that most of them express MAP2, VGET1. This is the day 14 time point post-iPSC induction. You can see very nice homogeneous expression for MAP2, VGET1, GAT2, and you don't see any other non-GABAergic markers like your glutamatergic markers, your dopaminergic markers, and so on and so forth. So again, very much like the glutamatergic, a very nice pure population of one cell type, in this case, the GABAergic neurons. The next CNS cell type that we have on the horizon will be the first of our glia subtypes, in this case, the oligodendrocytes. This cell type will harness a newer re-engineered version of our Optiox system, V2 design, which is version 2, designed to increase the transgene expression. And we found that this higher transgene expression is very, very important for making oligodendrocytes. In as little as 14 days, we get a very nice pure culture, homogeneous culture expressing with high expression of your classic oligodendrocyte markers, O4, MBP, and PLP. And all this occurs in as early as day 14. Now, for anyone who's familiar with oligodendrocyte differentiation, your standard protocols, they can take anywhere between 2 to 3 months. And it is very, very difficult to reproduce. It's notorious for that, in fact. And our system is able to, overcome those kind of limitations and produce these nice cultures, nice branch morphologies in as early as 14 days in the hands of the user. And we don't want to stop just there. We are going to use our cells to design accurate disease models as well. As mentioned earlier, alongside, we also want to make, alongside our wild type, we want to make healthy lines, but also we want to generate disease lines, such as isogenic models, where we, using the latest in gene engineering technology, we can introduce single mutations for genes such as tau to study Alzheimer's disease, for instance. But for a more faithful representation, we can also do human patient-derived models where we can, where we'll be able to take into factor all the different variation that can exist between one patient to another. And it's also very useful for diseases where you have more than one mutation and the disease is very complex. So this is, for us, these are what we are also working towards at bit.bio. Again, leveraging on the advantages of Optiox, we want to produce consistent and scalable cells, cell models of for human diseases, for research and drug discovery. An example of this using our first cell type on the market is the glutamatergic neurons. We are generating some of the most relevant diseases, such as Huntington's, Parkinson's disease, Alzheimer's. We also want to do this for the myocytes, to model disease, common diseases such as muscular dystrophy, mitochondrial diseases, and diseases implicating the neuromuscular junction. We also recognize the need to generate co-cultures. Things like the organoid is very popular these days, but with a very pure culture of cells, we've leveraged on 3D bioprinting actually to generate nice co-cultures of our neurons with astrocytes, allowing you to have very nice control over where you put the cell types. So here, with the renowned lab of Professor Hagen-Belly at the University of Oxford, we generated these nice 3D printed spheres where you co-cultured with human astrocytes. So you have human astrocytes in GFAP, and you also have here, and you also have our neurons in red. Again, to show you how using our cell types, the possibilities are endless. Going beyond just the 2D culture, we also envisage it for modeling 3D environments that are more representative of the human environment in vivo. As we have discussed, we have the GLUT, the GABA, the oligos, the myocytes, and so many other cell types for the CNS. But our aim is to do so much more than that. And we want to, alongside this, we want to generate disease models as well for pertinent diseases such as Huntington's and Parkinson's. And this brings me to the final slide here, where I would like to finish, to reiterate that, in addition to the CNS cell types that I discussed earlier, and in addition to the myocytes, our mission is to produce the next generation of human cell models for all known cells in the human body. And we're doing that by deciphering complex biological data, which grows by the day in this age of big data. And we do this to understand cell identity better, and using our OptiOx system, control cell identity to produce high quality, consistent human cell models at scale to support life science research and therapy. Thank you so much for your attention. And yes, this is our team here at bit.bio. And it's so much of this is thanks to the wonderful, fantastic, talented team that we are building here at bit.bio. Thank you so much, and I will we'll be very happy to take any questions you have.
So thank you very much for your presentation. It was very impressive, and the technology seems to be very promising. Now we have some questions in the chat. One is, first of all, very nice pictures. I have a question for Abcam. Do you have any multiplex kit for immunohistochemistry on brain tissue frozen or FFPE samples?
Sorry, can you say that question again? I didn't hear it at all. Please.
Yeah. It is written in the chat, and it is for Abcam. Do you have any multiplex kit for immunohistochemistry on brain tissue frozen or FFP samples? I mean, it is for Marie.
I think I understood the question.
I can take that question. So it's a very good question. We definitely have a wide range of solutions to support multiplex IHC. So we have different options for you to do your multiplexing in both immunohistochemistry, but also immunocytochemistry. So one option is to look for primary conjugates. So we've been, in the last few years, we really have been expanding our range of primary conjugates. These are already ready to go for your multiplexing experiments. And we try to really focus on our offering of Alexa fluor dyes because they are very bright. So you're likely to also get a very good signal for proteins that might not be as expressed as the highest abundant in the brain. So we have this conjugate range, and we do the validation in both IHC and ICC for this primary range. We also have, if you want to do that conjugation for a product that you don't find in our catalog, we also have the custom route where we can work with you to develop the conjugates of your interest. On a third option, we also have, for all our rabbit monoclonal antibodies, we provide them in different formats. So we have the carrier-free versions of all our RabMabs, which you can acquire and use together with our Lightning Link kits for direct conjugation. And that's a very sort of straightforward process to do the conjugation. And if you have the carrier-free version of antibodies, you can move forward and do that multiplexing. So really a wide range of options, but I would be very happy to discuss those in more detail, as well as to also forward your question to our product manager, who's actually Italian, Elena Locke, who could also address some of your questions.
Thank you very much. Now we have some questions for bit.bio. First of
okay, thank you very much. I can see some of these questions.
The microphone, so there are some noise. Okay. Very interesting, the new OptiOx technology. How does it improve reprogramming? Where are these cell lines shipped from?
Okay, can everyone hear me? Katrina, can you hear me okay?
Yes, we can hear you.
Excellent. So what I'll try and do is I'll try and the first point was made by very interesting technology. What other cell types are available? So as you can see, we have our IO glutamatergic neurons, which are available. This is our first cell type that is available. We also have our IO skeletal myocytes. We have a product development plan in place to provide multiple cell types, which include a comprehensive collection of cell types involved in CNS. So as mentioned by Kaiser, we have cell types in terms of IO GABAergic neurons, IO oligodendrocytes, and we have also planned product development programs in terms of dopaminergic neurons, as well as motor and sensory neurons and microglia and astrocytes. We have a program over the next 12 to 18 months to develop and deliver the best-in-class neuronal cell types, as well as other cell types, including immune and hepatocytes, as well as other in disease areas. So right now, you can order directly from bit.bio or through Abcam the glutamatergic neurons and the skeletal myocytes, as well as the wild type parentals, as mentioned by Kaiser. We're making disease models. So we have CRISPR technology within bit.bio, and we can engineer knock-ins, knockouts, point mutations, and SNPs to help model disease as well. And so we have active programs with multiple partners that allow us to do that. And we're very open to collaborating with other companies as well. So I'm just looking at another set of questions. I would like to ask if Optiox can work with iPSCs brought from some other company. The answer to that is yes. We have a comprehensive parental family of iPSCs which we use, and we have total freedom to operate to use these iPSCs for our own commercial products, but we are open to working with other iPSCs as long as the correct freedom to operate licenses are in place. Another question by Raffaella Ravazzi. I would like to study neuromuscular junction. Can the IO skeletal myocytes be used? Yes. In the presentation by Kaiser, one of the slides specifically mentioned the fact that you can study neuromuscular junction. The IO skeletal myocytes, you can see that they are contractual multinucleated myofibers, where they show an expression of dystrophin and glucose transporter, and they respond to neuro-induced stimulation, and they can be used to study neuromuscular junction, metabolic disease, mitochondrial diseases, and muscular dystrophy as well. And the final question, I believe, is how are IO skeletal myocytes better than primary skeletal myocytes that are currently available? Well, first and foremost, Kaiser, do you want to take that question on first, and then I can add my part?
Sure. Yeah, how are they better than the primary cells? Well, in this case, it's the best part about it is that you have control over the cells, and you have over things like the cell numbers that you have, starting with the seeding density. It's much easier in that sense that you can control the cell numbers as opposed to working with the tissue. And it's better also in a sense if you can for studying development compared to maybe, let's say, working with an end state disease, end state model. And of course, being a cell line that you can easily obtain off the shelf, it is easily available and it can be easily scaled up compared to, let's say, primary cells, which really just depend on the source that you're getting it from. Kam?
Any other question?
Just to add, on behalf of Kaiser, we are not providing cell lines, we are providing primed cells. So in answer to that particular question, why are they better? Primary cells have struggled in a drug discovery environment, especially in terms of their amenability to culture. They're difficult to maintain. You can see from the information that was shared as part of the reprogramming strategy where we use MyoD1 that the cells are easy to revive, that they remain highly characterized, they follow a set protocol in terms of reprogramming, they are easy to maintain, and they can be used for a wide array of metabolic studies. They are easy to culture. So there's a number of factors that come into play when you compare them to primary myocytes. Okay, that's it.
Both neurons and skeletal cells are available in the Abcam website. So if you want to have a look, you will find more information also for the commercial point of view. Okay. It seems that no more questions are coming. So thank you very much for the participation to this webinar, and feel free to send us your question if you have more things that you wanted to know. And thank you to everyone for attending. Bye.
Thank you, everyone.
Thank you all. Thank you.
Thank you.
Thank you very much, everybody. Thank you. Appreciate the opportunity. Goodbye.
Thanks.